Good morning, I'm Mr. Cuccherini. Here we are at J.R. Tucker in today's classroom, and this is my IB biology class, and we are reviewing cell biology and magnification. All right. So, we all drew our eukaryotic cell, correct? Now, what are the other two types of eukaryotic cells besides animal and plant that you could have drawn? What are their categories? Eukaryotic. Come on. Fungi, good. Fungus and protists. I heard protists, good, protists, very good. All right, now the next thing I asked you to do is this magnification question. So if we had a blue whale drawn on our boards that was 10 centimeters long, and its actual length was 50 meters, which is a lot bigger than an actual blue whale, but we're just going with it for ease of math. What would the magnification be for that blue whale? So remember, magnification was measured over actual, right? So if actual is 50 meters and the measured is 10 centimeters, what's our first step? Right, convert the units to make them the same. So I think you already did this, Philip. So what is 10 centimeters converted to the proper units? You just did the math like a robot in your head. Anybody? 0 point 0 point, so it goes by 100, so it's 0.1, right? All right, and then if you divide 0.1 by 50, what do you get? 0.002. Right, so then it equals 0.002x. And that makes sense because a blue whale is actually being shrunk to fit on our board, whereas most of the cells we draw are being blown up. So you'll see them magnified by like 1,000 or a 10,000 scale, whereas the blue whale is actually being shrunk, okay? But the formula is what you need to remember, measured over actual, and you also need to remember to convert your units. So this is 0.1 meters divided by 50 meters gives us 0.002. All right, are we good with that? Yes?
For the first part, even though it's centimeters, why did you divide by 100 instead of multiplying by 100?
Why did I divide this by 100?
Yeah.
Because I'm converting centimeters to meters. And how many centimeters are in a meter?
100. Okay, so you divide by 100. Okay.
So you divide by 100. Good. All right. Next one that we're going to review, you guys can clear your boards. Oh, shoot. Distinguish between eukaryote and prokaryote cells. So draw on your table any differences between eukaryote and prokaryotic cells. And I'll let you guys decide. How many marks is this going to be worth? Not one. If you don't pick quickly, I'm going to pick a number. 12? All right, we'll go.
I'm sorry.
I'll give you 5. How about that 5? We'll meet in the middle. Well, so remember with distinguish, you have to have a distinguishing remark for both categories to get 1 mark.
They're more basic, like.
Oh, come on, guys. Oh, Miss T is going to be so disappointed.
Can they both move?
I should.
So what else would we put down for the prokaryote?
Those can technically be on both.
Me too. I think so. We know this is bacteria. We know that it's I'm screaming. We know that it's unicellular. Sometimes they have like flagella. Yeah, well, that could be like that comes between them. Okay. So that
Oh, I know what you're talking about. Okay. Now
Isn't that this one is just because like this one has the ability to be like multicellular, like it's a common occurrence.
So like we can.
In terms of
okay, so also think about the way they
So there's something
reproduce.
cellular, right? Like a bacteria cells is unicellular, while eukaryotes are multicellular.
They can be, but some eukaryotes like amoeba.
I know, but I'm saying this is the ability to be multicellular. This one can be multicellular.
A specific way that we phrase it for prokaryotes.
Don't they make like a copy of themselves?
Yes.
So, Monica, do you remember?
All right. Reach in your brain. I'm sure you can get it. Keep trying.
Okay, thanks.
All right. It's not the same as how eukaryotes do it when you go through the whole cell cycle. Remember all those phases?
Yeah.
Those don't apply to prokaryotes. So
They
we
just
have
make a copy of their
Right, so-
own DNA.
so we have a name for that.
I
We have a
know.
couple names, and I'll accept any of them if you can think of them.
All right.
And then the other way applies to eukaryotes. So if you can remember the name for that. All right. Okay. You guys are thinking about the DNA. That's good. Folded DNA. All right. No. But what is it folded around? Can you remember what it's packaged around? Those molecules.
We'll get to it.
Making you read all the way back to last year. I know it's painful. Like this is too confusing. Okay. All right. Yes.
Vacuum, right? Those are plants. Those are your eukaryotes. Simple versus complex. Well, that's a little broad.
Are you talking about the organelles? Okay. Yeah. So it has to do with what surrounds them. Histones. Good. Ask me what. Oh, okay. That is a very specific term for a specific type of cell. And it could count depending on how the question's worded, but I'll give you some more basic ones. Okay? All right. So, what is one of the immediate differences you should remember for eukaryote versus prokaryote? All right, so multicellular versus, all right, so most or a lot of eukaryotes can be multicellular or unicellular, depending on the kingdom, depending on the organism, but prokaryotes are always unicellular, single celled. But what about the nucleus? Right, so pro means no nucleus, eukaryotes do have a nucleus, right? So that's some of the basic stuff. Inside the nucleus, for the eukaryote, you would have DNA, but how is the DNA packaged? It's wrapped around histones. Good. So the DNA is wrapped around histones. So you have DNA associated with proteins, so wrapped around histones. In prokaryotes, it is free-floating, or you can call it naked DNA. You're allowed to write that. And it is not associated with histones. What is another form that the DNA can take? It's that circular form that can be passed from one bacterial cell to another. This is a what? Oh darn! So like when starts with a P. So this is going back to when we were talking about antibiotic resistance. If a bacterium has a circular piece of DNA with the trait for being resistant to an antibiotic, it can actually just copy that and pass it on to its friend. And that's how they can spread that ability rapidly. So it starts with a P. It's a circular piece of DNA called a plasmid. Plasmid. And some people have some bells ringing. All right, so to help you out, we'll go ahead and look at all the answers. And then I'll see if you guys have anything extra, but make sure you have all of this written down so for practice. All right. So organelles, prokaryotes, no nucleus, no membrane-bound organelles whatsoever. Here, the DNA we said is free-floating or naked. Circular is referring to that plasmid that I was talking about. DNA is linear and it's associated with histones. That's what the protein is. Remember, for eukaryotes, the DNA can have introns or exons or coding regions versus non-coding or regulatory regions, right? And that's not so much the case for prokaryotes. When I say 70S versus 80S, what is that really referring to? Yeah, basically the size, the molecular weight. So you don't need to necessarily even know what the S is standing for, other than the fact it's like a unit of measurement associated with mass and size. All right. So prokaryotes, a way to remember this is they are smaller, simplistic cells, so they would have these smaller weights. 70S ribosomes, 80S ribosomes for the eukaryote. All right. And reproduction. This is the thing I was trying to get you to pull out, Rowan. They go through binary fission or cloning, because bacterial cells they don't have to necessarily copy every piece of their DNA, and they don't go through all of those cell cycle phases we discuss with eukaryotes, like, mitosis and meiosis. All right. Now the size. This is a big one. You have the average size of a bacterial cell is about 1 to 5 micrometers. Eukaryotes, it's 10 to 100 micrometers, and there's obviously exceptions. All right. Most of them fall into that range, though. If we were talking about viruses, where would they fall? Very small. So do you remember or a unit of measurement smaller than micrometers that you might have talked about? Nanometers. Good. So nanometers would be like the viral scale. Okay? Good. Yes?
Is there something that has to do with like finding food or making your own energy?
Oh, good. So their mode of energy it's not a good point to distinguish the two, because depending on the kingdom for eukaryotes, they can be autotroph or heterotroph, and the same goes for bacteria. They can be autotroph or heterotroph depending on which one you're talking about. You could, if you were doing a compare and contrast question, so reviewing our command terms, that would come up, because then you could talk about that as a similarity. But for this question, remember when we say distinguish, like this whole row here is your single mark on that final test, okay? So you can't just talk about one and not the other. You have to have two paired statements when you're distinguishing, all right? Good. All right. This next one hopefully isn't as challenging, but let's see. I know, I purposely picked all the stuff from last year. We're starting off with that. All right. So we just talked about cell division a little bit. So this is a eukaryotic cell. Remember how I told you to represent cell membranes?
Double it up.
Yes, double it up. There you go.
Wind up in the middle. Oh yeah, you're definitely right. That is what it is.
That's the
Want to get a new marker?
You have the mitotic spindle, though, to be more specific.
Wait, what did I? Oh,
And then, what are these conjugates?
I
Those
said
are my
point
spindle
one to you
spindle
guys,
fibers.
right? In that
Four
last
pulling them
example,
across.
I wrote point one on the board.
That's
I thought I did. All right. I hope I did.
What's it called?
I understand. It's right before Easter. Everybody. Metaphase.
What are these things called?
They line up in the.
What are the little hairs called?
Spindle fiber?
No.
Everything else looks great. Everything's wrong. Oh, yours looks good. You're missing what goes here and here, though. Yeah. Let's go over it. Let's assume that it was a 5-mark question. All right. So you could have labeled your cell membrane. By metaphase, what inner membrane has disappeared already in order to allow you to move the chromosomes around? What are they contained in a eukaryotic cell? A nucleus. So the nuclear membrane has dissolved. Good, good. All right. Each chromosome is technically paired with it's a pair of sister chromatids. So this band here is an identical copy to this one over here. All right, metaphase, they line up in the middle. And just for review, mitosis, what are the phases? PMAT, good. Prophase, metaphase, anaphase, telophase. Cytokinesis isn't technically part of mitosis for what you're talking about, all right? Now, we'll discuss all of them in a second, but anaphase and telophase, telophase, however you want to pronounce it, is fine with me. The centromere is a protein complex that allows the sister chromatids to bind to each other, and it allows what you might have called spindle fibers, which is it's the same thing, both count. So if you erased it, you didn't have to. But the microtubules, spindle fibers, they're like fishing line that attach to the centromeres and allow these centrioles to pull and separate the sister chromatid pairs. All right? Yes?
So on the IU exam, would you write microtubules or spindle fibers?
Both would be counted, I would hope. Spindle fibers and microtubules are acceptable because spindle fibers are a type of microtubule. And in most keys, I've seen them include them both as an appropriate answer. You could say the most specific one would be spindle fiber. All right? Yes.
If we get the question outline mitosis on the exam, do we include cytokinesis or no?
Don't.
Okay.
Okay. Now, I mean, that's again with IB, they won't count it against you, but it would be wasted space. So if you go through everything and you had it at the end, everything else is right, you'll still be fine. All right, but cytokinesis technically isn't part of mitosis. Mitosis is about separating the nuclei, essentially. Okay? Good. Any questions about that? Nope. All right. Thank you for joining us today. Hope you enjoyed our lesson on cell biology.
Go Tigers!
All right.